Hello, everyone. So thank you to be here. Today we have Pau from Cambio Global, a PhD student, to present his study. So Pau, welcome, and
Thank you.
it's a pleasure for you.
So I'm going to talk about the last chapter of my PhD thesis, well, my first chapter, in fact. But first of all, we are going to do a short introduction about pollination on islands. So we are going to talk about why it matters, the pollination. We're going to talk about the pollination crisis, why we studied on islands, and plant pollinator networks, some basic concepts to understand my first chapter. And then we will go into my first chapter. Okay. Well, pollination is the transport of pollen from one flower to another. This process can be by the wind for some plants, and there are different processes, but we are going to focus on the pollination by animals. So in our imaginary is the honeybee, the queen of the pollination, right? And because we obtain honey from this species, but this species is considered invasive when we study wild ecosystems because it replaced the native pollinator. Okay, there is a huge diversity of pollinators in species, in shapes, and each of them has a different role in the ecosystem, and they pollinate in a different way. We have butterflies. This is a very small bee. Also in bees, we have a lot of different species, 20,000 species in the world. We have flies also, wasps, beetles, and also vertebrate pollinators as birds and lizards. And also recently, they didn't check the efficiency, but I mean, when we talk about pollinations, we can expand our minds. It's the transport of pollen from flower to flower. Okay? So it matters also not only in wild ecosystems, but also in crops, because 75% of the crops of Spain depend on animals for the pollination of the crops and the production, and some of them cannot be pollinated by the honey for the honeybee. For example, the tomato flower, you need a bee that does the past pollination, is a kind of vibration to open the flower and obtain the pollen. Or for example, for the avocado, only flies can pollinate this fruit. So the honeybee cannot replace all the pollinators. There is a global crisis. My supervisor, Ana Traveset, wrote a book. It's quite well known. There is a decline of the population of the pollinators, and they have an economical impact. That is the only way the humans understand to preserve the species. So they are worried in Europe and around the world. So the solution is in our hands, and the solution could be constructing bee drones to replace all those pollinators and all this diversity. And in fact, is a solution. So problem solved. It's a joke. Why we study islands? We study islands because they are natural laboratories. We can have different fragments to study different situations. For example, having an invasive species in one island but not in the other. So it's kind of a laboratory to study this, or study extinctions. For example, in Ibiza, some native species are. Where is the nose? When there is an invasive species, the propagation in an island can be very quick. And in some years, the snake arrived to Ibiza and disbanded in almost all the island. And now the Podartis, the endemic lizard from the is only in this area, and in this area is sharing space with the snake, but it's going to disappear. And all this area it disappeared in 5 years, so I think it will be extinct soon. So, plant pollinator networks. Some basic concept, we have two subsets, plants and pollinators, and they can have different ecological roles. Like for example, generalists are those ones which interact with a lot of species. Specialists, like this plant, interact, it has only a few links in proportion of the networks. Then the nestedness is the proportion of specialists interacting with generalists. Modularity are submodels into a network that they can be more or less connected. And the connectance is the proportion of the links that occur to all the possibilities in the network. So robustness we can interpret this to like the robustness to extinction. For example, if we take out one species, so another one can be extinct if no one pollinates this species. So you can simulate removing species, and you can know when it collapses the network. Identifying keystone species, those generalists that they can connect different modules, so they have an important role, and they are important for conservation to maintain the structural properties of the network, and interaction gaps. For example, is an opportunity for ecological restoration. If one species disappears, we can provide another one that has a similar one. So, yeah, for the stability of the network, we need sufficient modularity, but not to a point that propagation from if we have an infection in some module to not to be as connected to propagating the whole network, and a moderate level of redundancy, because if we lose some node that provides the link, so someone can replace that node. So generally, sometimes are important because if some species disappears, so someone can replace it. So what we have to do is preserve what we have, and it works, not to put drones around the world, and ensure the structural properties of the network to face the global change, which is the reality. So now we are going to get into the first chapter of my PhD thesis. Is a comparative assessment of automatic and manual monitoring in comprehensive plant-pollinator communities. So as we were talking, there is a worldwide pollination crisis. The plant-pollinator community studies are focused on diurnal data because we are diurnal and we take the data from diurnal. And traditional methods have some limitations like strength, or some study sites are remote and difficult to reach. So yeah, we want to obtain a bigger data set to have bigger resolution in our networks, and have a permanent register and analyze those interactions in detail. So automatic monitoring has some benefits, like it can be in the field longer periods, it can obtain nocturnal interaction from the whole night, census without human presence, yeah, and to obtain this, we have to obtain first a pool of pictures, then we have to tag all those pictures and train and obtain an algorithm to automatically detect those pollinators as a camera trap. So at the end, what we have is a program that classifies the frames in presence or no presence. So we used a single board computer with a power bank supply for the energy, and a camera with a focal length with a 3D printed inner case, an external case to protect it from environmental conditions, and we obtain the data. We obtain the data placing those cameras in the field and performing focal observations. And we obtain a network with the cameras and with the humans. And we tested how different are those data. We did it in Cabrera Island, a small island in the south of Mallorca. And yeah, we had 6 study sites. We did 13 field campaigns between March to June from 2022 and 23. We were 2 field technicians always in the field, and we placed 2 cameras per plant species, all the plant species pollinated by animals in the community. So we tested the metrics we were talking about, like connectance, specialization, robustness, and also the species richness, how many species obtain each method, and the abundances of those species. So the algorithm we trained, we obtained high accuracy with cameras of high quality, which has high quality. And we manually reviewed all the videos to test if the algorithm fits on what it's saying, and we obtained an 86% of accuracy in detection, not classification, only in detection of pollinators. So here is an example of a video. Okay. So we can see.
Aqui vais, podes passar.
Okay. So we can see that the algorithm can detect pollinators independently of their size, like a bumblebee, a Coletes bee, which is quite small, or a Microdiptera. But also it does some mistake, like it recognizes as a pollinator sometimes, what is not. So when we check the both datasets, we obtained similar species richness and Shannon diversity, which is quite good because it means that both methods are agreed among them and are quite reliable. But when we have in the x-axis all the pollinator species, and the flower visits within 6 hours, we obtained they were more detected by the automatic monitoring. When we check this per plant species, we obtained the same by the number of individuals, but the pollinator events, for some plants, they were recorded more by the direct observations. This could be because in Cabrera there is a high abundance of Podactyl lilfortis, a lizard, and when we are doing the census, this animal is more shy and pollinates less, and its presence can make that pollinators don't go so often to some plants if it's quite frequently pollinated by those lizards, because they also eat insects. So we find some differences in the connect times, the specialization of the network, and the robustness. This could be explained because as the cameras stay longer in the field, they stay more time recording interactions, they obtain more sporadic interactions, more low frequent interactions. Then it increased the proportion of the links between all the possibilities. So the connectance is higher with the automatic camera system and lower with the observation. And the same happens with the specialization, because those sporadic interactions are like specialized, they are interaction very specialized. So it increased, and also the robustness, because it increased the connectance. So the network, it seems to be more robust if you obtain those interactions from the cameras. So here we represented the network. Here we have the pollinators sorted in the same way, and the plants also sorted in the same way. So in general, we can see the same pattern. Well, sorry. The same pattern. And as more black are the square, more interactions are detected. So this is a frequent interaction in both networks. So the general structure is the same, but here in red we have the missing interactions that each method has done. Okay, and the camera has done more missings than the humans. It could be explained by the lizard. When we check the missings of the direct observations, we plot it, and we can see here we have two y-axis. We have the number of overlapping points here and the frequency of interactions. So when the frequency of interactions is 0, is here in this axis. And when the overlapping points are, for example, for one interaction, if it's missed 30 times, it's here. So as less frequent are the interactions, are less captured by the direct observations, confirming what I was saying. And as more frequent are the interactions, less missing is performing the focal observations. So when we check the missings of the automatic camera systems and we plot it, we can see that we have a limitation on the size, because the smaller pollinators are they presenting more missing with the automatic camera systems. So there is a size limitation in this method. So take home message, the automatic camera system can present a balanced data set which is not biased, because what happens in the network is when we are not there. And it obtains more low frequent interactions, those sporadic interactions, more exclusive interactions, but it has a limitation on the size of the pollinators. I will put it here. And yeah, so they are valuable for monitoring those communities, and yeah, and we test it. So this is a video of a lizard pollinating. Okay. And yeah, future works. I'm going to work with functional groups classification. We started a bit. Not only detecting the pollinators and having to check manually on all those interactions and take notes manually, and also with nocturnal, like detecting nocturnal pollinators and have a broader network. And also we have data with my colleague Laura. We took data from some islands of the Seychelles where we have this invasive species we were talking about, and islands without. We're going to test it also in the Mediterranean. And yeah, with Aranchalana, that is a technician from IMEDEA in the data lab. We did a course here in IMEDEA. I think we are going to repeat it. You are all invited if you want to attend. How to train an algorithm for detecting fish or insects or whatever. And using those Raspberry Pis, that they are the small computers that I have used for programming the videos in the field. And that's it. Thank you. Questions?
I have two questions. first one in the results, you say that there are many more missing interactions when using
Yes.
the automatic detection. If I understand correctly, these missing interactions, it's not that have happened, but it's that they are not happening and they should happen. Or how do you know they are missing interaction?
Because it's for the same plant and for the same pollinator, but not for the same individual of plant. I mean, in a community, you can have many individuals of the same plant species. So at the end, you are collecting time. I mean, you are collecting time. You are investing time on that plant, for example, no? And you are obtaining interactions for that plant with focal observations and with the cameras. And when you compare it, you can have missings
Because in the other.
because the other method collected those interactions.
So you are assuming both methods when are.
Both
having
methods
some
has
result, it's a good result. It's not a misunderstanding from the person or the camera. So you're assuming that both are true observations.
yes, because occurred. So there were interactions that happened because someone saw it and took
Over the
notes.
cameras, but you are.
And the
assuming
cameras.
That the camera also won. So the camera was true and. Do you understand me? Somebody has.
Yes, it's true because we check it.
Every single interaction that has gone through the camera.
Yes.
Somebody has checked.
Yes.
Every.
Yes, me.
So everything.
A lot of
is
time.
true.
Yes.
You're assuming that the person has not missed any interaction.
Well, I assume because in the notes of the
Yeah.
technician, this interaction it doesn't
But
exist.
some others could be missed, and you understand that those are the ones that the camera observed. No? I mean, you are assuming in both that it's true. What the camera has, or what the network has seen and the person has seen, it's true. And what they have missed is because they have just missed the person and the camera. It's like that? There is
Yes.
Okay.
Yes.
Okay. And
No los convences, pero sí.
right. And the other one, I have it in my mind. It's difficult, so I will tell you later.
Maybe this one? No, I don't know.
No. Well, I have one thing to say that maybe you could improve when there's the automatic detection is seeing a flower. Maybe you should introduce this class.
Yes, the flower.
Yeah.
Yeah, it's the next step totally.
If you
To
some flowers inside.
to detect
If you
the
go-
interaction of not only the pollinator. Yeah,
And then
this is necessary. Yeah, totally agree.
I had a curiosity about how you plan the cruises or the campaign. The planning of what you're going to sample.
Yes. When we choose a site to sample, in Cabrera, for example, there weren't a lot of plant species because it's a quite simple ecosystem. And when we choose we make a list of the plants that they are flowering in that moment, and we put 2 cameras per plant species. And for the rest of individuals, without trying to disturb them, we were doing focal observations.
Yeah, but how to decide where to go for islands, so we choose.
Because for another project we had like habitat. We want to study different habitats of Cabrera, dune system, rocky coastal, and pine forest, and we had 2 pine forests, 2 rocky coastal.
Every time the same.
Yes,
Exactly.
yes.
Perfect.
Yes, Sarah?
Do you have a way of knowing if the fact that the camera is avoiding insects or other animals or things go into the flowers?
What? Sorry, I mean,
So
repeat it.
and what I mean is, you have the camera in front of of the plant.
Of the plant, yeah.
So is
Start.
there a way of knowing that maybe you are missing, or maybe it's affecting how often pollinators are doing because there is something in there? You know what I mean?
Like if the camera disturbs the, yeah,
The
maybe
normal-
maybe can disturb
So,
because of the whole
Even
as the person. Yeah, I mean, I'm not saying that camera doesn't has a bias.
no, but I'm asking if you know a way of testing and kind of like quantifying how much it is affecting.
It's very difficult because the way of testing if humans has an intervention in my imaginary is with cameras. But how cameras disturb this? No, I don't know.
Not just thinking if someone had it done, maybe in the past. I don't know.
Maybe I will check it.
Would be nice to.
I will check it.
really related to that, how far is the camera, like how far away from the plant is it?
Yes, with is 40 centimeters more or less,
Okay.
almost half a meter, because the camera has a focal length which is a 35 millimeters, so you can put it quite far away and have a good picture of the, well, as you saw in the video. Yes, Marco?
Actually, was following up on this. I was wondering how you decided the magnification to use, because if you have problems in
Small.
detecting and classifying small insects, then that would be right. Moving would make you move more towards a higher magnification, but then you lose other things, so things get out of focus. And
It was a threshold between controlling, because there is another fact that humans can control more number of flowers than the cameras. So it was a threshold about controlling some kind of flowers, I mean at least some number, and yeah, losing some pixel definition for small insects. Go ahead.
Do you program the camera to autofocus when it detects something, or that is automatic?
No, there is cameras with,
An autofocus thing.
there is autofocus like our mobile phones, like they can do autofocus, but the cameras I'm using are focal lengths and I have to focus manually every time. And yeah, I plug the screen in the field and I focus it. Camera by camera, only one screen, plugging into. Yes, more good. You.
I remember it was about
Yes.
the size of the
Hmm.
pollinators, that maybe it's related to the network itself, because I imagine you type what is the size of the image, it's like 1,200
Mhm.
pixels for,
Mhm.
no?
Yes,
And probably
exactly.
not go to 600 or something like that.
Exactly that size, exactly.
So you are losing the smallest one because you are losing definition inside. So you probably should try also, which is longer, but you should try to train with the bigger size of the image. That you can do it. I don't know how many images you have,
Okay,
but you
okay,
can try to work with the bigger image so you won't lose the definition of each pixel.
okay. I will try. Yes, Marco?
yeah, I was I was thinking maybe you might also want to look into cameras that are used for machine vision, like telecentric cameras, so telecentric objectives, sorry, that essentially allow you to not lose focus.
Okay.
You lose a little bit of resolution, but you don't have this problem of putting in focus. Yeah, if you want, I mean, I just tell you the name, and
but-
you can check it up and
Okay,
look it up and see whether it might.
but
be
it focus automatically or?
No, it is something has a very long.
Okay, and you can put it more far away.
Well, the idea is that then things will not get in and out of focus, essentially.
Okay,
They're called telecentric objectives.
okay, interesting. I will check it.
Yeah, check it out.
Okay. So yeah, for the lizard, we've had many interactions that, for example, in human observations, we had like one lizard interacting with 3 flowers, and in the cameras, we had 5 lizards interacting with 5 flowers. So the difference was huge in the case of Cabrera. There are no lizards in all the island, and the difference with the lizards was quite huge. Yes, Enrico?
Yeah, so here regarding this plot, because the missing links, so do you attribute them to this, like that you are not able to identify or see the insects on the videos, or because I was wondering if the sampling effort was the same with the cameras than the person, because I said that the camera is just looking to one flower, and maybe when you are looking, you can look several more flowers, so your effort is bigger because you are sampling a bigger area. And then, so I was just wondering that if multiplying the number of cameras, do you think that you could achieve the same?
Yes, I think it's a matter of number of cameras, or it's a matter of how many flowers do you control.
Okay.
Because of course, if there are we are 4 in the field instead of 2 people, we will have like more interaction. Maybe the difference is more. And if we had more cameras, like the double of cameras, we will have I mean, it's a matter of how many flowers you can control.
Okay. Because here
and the frequency of visits that they have.
In the human part, is you put all the observations you have. And you could try just to put the same number of flowers you have with cameras to see if they.
Well, we control it. We control it in the representations of here. We control it, the number of flowers and the time invested by each method. Kike?
Yes, I was distracted, but I was thinking now. So, do you do like an standardization with the time invested somehow? Like when in the effort you are investing, do you consider that at the time of calculating the index from the different methods? You know, like in fisheries, you have all your fish, but you consider the time or the effort in different ways that you are investing in fish that to calibrate if how that represents the population. So I don't know, in this is something similar when you calculate this index. It's not the same to have 5 identifications in 5 hours than in 1 hour. No, I think, I don't know. Okay. So I was wondering if that is somehow considered in this kind of index, or it's just a characterization, not a quantification, what it's based on.
It's a quantification controlled by the time invested and in flowers control, and that's it.
Good. So it's okay.
Yes, it will be good.
Okay.
The algorithm you used, it was a machine learning one.
What?
The algorithm you used to detect the bee, was it a machine learning one?
Deep learning?
Yeah.
Deep learning algorithm.
How did you use enough sample for it to be efficient? Because it needs to be like a lot.
2 years of sampling.
Only you take like the data of all this to feed the algorithm.
To feed the algorithm? Well, we use also image from internet or from cameras and this, but sometimes it doesn't improve the detection, because what is better is to use to train the algorithm with the same kind of image you are going to work with, because it has a different depth of field, and yes. That's it. It's important if you want to train something, to train with the kind of image.
Have you ever compared the detection that you get? I mean, now for person when you go with other technicians or other students. So, because for instance, myself, I'm not trained at all to do this.
Yeah.
Use everything, but so there might be people who observe better than others.
Yeah, there is a thing here. We didn't not explore about this, but it's true that some field technician can have more experience, or for example, me, I have more experience now than in 2022. It's a fact, like when someone new comes with me in the field, we try to do the first census together and yeah, do like a training to, but yeah, of course, between people there is differences.
But that would be applied also to the scenario when person is installing the trap, right? Like the camera. Like also can be biased the same way.
But it's always, yeah, but it's biased if it's always the same observer. If it wasn't a different observer, you could lose, and if you had like a lot of samples. But if you, I think there is a problem, and the consistency of the automatic detection all along the data set, it's much better just because of that, because it's like really consistent on what is.
At least this is standardized, yes, because it does it always the
If you're
same.
going with different observers, you don't know how biased is it.
Each observer. Yeah, one of the results we saw in the data is like humans tend to observe more times what is common, what is easy to see. And what is sporadic is more detected by the automatic camera systems. But maybe those super frequent interactions are well captured with the cameras, but not as much as humans. So a lot of missings from the cameras were that they got those frequent interactions, but not as much as humans.
But because the problem of the number of cameras, not because.
Yeah, a number of cameras
If
or
you normalize.
yeah, it's true.
Instead of comparing 5 flowers with 1 flower. if
It
you
could be a
normalize it,
human bias
you could
or
see
also
it.
it's a matter of number of cameras. Yeah. At least what is interesting if, for example, if you are studying a difficult to reach site, site which is difficult to to reach, um, to put also those cameras, it's expand your network resolution and your data because it's a different kind of census and it provides you a slightly different data and yeah, maybe you can remove a bit of human bias and add some camera bias, but maybe it's more, yeah, the picture is more realistic. Because even you have less interactions, you are controlling less flowers. So you have a picture of exactly what is happening in the flowers. And mostly you can check it after time, for example, in 20 years, or I don't know. You can check those interactions, how occurred. Or if there is pollinator interference, for example, if a bee is taking away another bee always, or another pollinator, or you can check all this information because when we are in the field, we are taking notes. And sometimes there is a lot of activity that you cannot be super precise of what happened exactly. Sometimes it's an estimation. Or there are details that you cannot take notes, you don't have time to, or they occur super fast and it is.
So we can leave it here.
Thank you very much.